Welcome everyone. This is Lynette Kraft, Chief Science Officer at ACSM. We're happy to have you join us for our first Brown Bag of 2019. The goal of the Brown Bag series is to introduce our staff and students to our members and the breadth of science that they conduct. We'll provide a brief introduction of today's speaker. We'll then turn it over to her for our presentation, and then we'll take questions following the presentation. If you have a question, please type it into the question area on the GoToWebinar screen. If you have any difficulty with seeing the screen or hearing the audio, it's best to log out and then rejoin the webinar. Today, we're pleased to have Dr. Erin Simmons with us. She will discuss ongoing research within the Warfighter Human Performance Program at the Navy Experimental Diving Unit. This research focuses on the physiological response to diving hyperbaric environment in an effort to mitigate any resulting decrements in performance that could affect mission success. A little about Dr. Simmons. Erin earned her Ph.D. in nutrition from Texas A&M University, where she performed research on muscle protein synthesis and sport performance. Following her master's degree from Florida State University, she worked as a laboratory technician at the Navy Experimental Diving Unit. At Texas A&M, her research focus shifted to nutrition and exercise physiology, a decision facilitated both by her time at the diving unit as well as her role as a volunteer assistant coach with the multi-year national champion Texas A&M track and field team. Currently, she is a Department of Defense contractor working as a research physiologist to study human performance and nutrition at the Navy Experimental Diving Unit in Panama City Beach, Florida. So at this point, we'll say welcome to Erin, and we'll turn it over to her. Again, if you have questions, please type them into the question area, and we'll take some questions at the end.
All right, well, hello, and thanks for having me. Like you said, my name is Erin Simmons, and I'm a Department of Defense contractor working for the Villaquia Research Corporation, and the research that I do is conducted in the Warfighter Human Performance Program at the Navy Experimental Diving Unit. This program is headed up by Dr. John Florian and falls under the Biomedical Research Department at NEDU, which is overseen by Captain Tom Robinson. So just to give you a little bit of information about me, I received my doctorate from Texas A&M University, and my dissertation project was interdisciplinary between nutrition and kinesiology. I received my undergraduate master's degrees from Florida State University, where I was also a student athlete, and I actually originally specialized in marine biology. However, after my first internship at NEDU, as well as working as a volunteer coach for A&M track and field, I ended up switching my focus to physiology instead. So like I said, I did my first internship at NEDU in 2011 after I graduated with my master's, and I stayed on a while longer working as a lab tech before going back for my doctorate. And then I also did a second internship at NEDU in 2016 while working on my doctorate, and now I am back working as a research physiologist. So at the end of this presentation, I will provide some resources for getting involved in military research in case anyone is interested. But mainly today, I'm going to focus on some of the research projects that I've been involved with over the years. But first, just to give you an overview, the Warfighter Human Performance Program conducts research with a focus on improving human performance, readiness, and mission optimization, primarily in extreme environments. Most of the research done in the program at NEDU falls under the diving or hyperbaric scenario shown here, which is the condition of higher pressure relative to sea level versus normobaric, which is pressure at sea level, and hypobaric, which is pressure less than that at sea level. So you can see here some of the areas of research that the Warfighter Human Performance Program has been involved in, including thermoregulation, diving physiology, biometric monitoring, and oxygen toxicity, and of course, human performance, which includes everything from physical exercise performance, hydration, nutrition, cognitive function, and sleep. And much of the ongoing research focuses on how diving and/or water immersion can affect specific outcomes, such as exercise tolerance and performance, fatigue, vascular function, orthostatic tolerance, muscular function, vision, pulmonary function, cognitive function, and nutritional requirements. Additionally, military diving environments can be quite variable, so the program has also investigated cold water diving, hot water diving, long duration dives, wet versus dry dives to look at the effects of water immersion versus pressure alone, air versus oxygen dives to parse out the differential effects of breathing gases, and resting versus exercise dives to determine additional impacts that working dives may have on performance. So today I'm going to go through a couple of studies that will touch on these different variables in diving environments. The first study I worked on as an intern at NEDU investigated the effects of repeated 6-hour 100% oxygen dry chamber dives on diver performance and orthostatic tolerance. And orthostatic tolerance, which is the ability to withstand hypotension during gravitational stress, can be an issue with diving and water immersion due to the stimulation of immersion diuresis and resulting hypovolemia. Symptoms of orthostatic intolerance are loss of postural control or consciousness upon transition to an upright position. So obviously, fainting or an inability to maintain posture could be a big problem for a diver emerging on land after a dive. So this study was conducted after previous water immersion studies showed a greater decrease in dynamic exercise performance and isometric strength after 100% O2 dives compared to air dives. On the other hand, orthostatic tolerance was diminished more following air dives compared to O2 dives. So the purpose of this study was to investigate whether dry O2 dives had similar effects as immersed O2 dives. And this study utilized 12 young, healthy divers, and you can see the summary of their characteristics in the table here on the right. 6-hour dives were performed in a hyperbaric chamber at 1.35 atmospheres absolute, and divers completed both a single dive as well as a dive week consisting of 5 consecutive dives. And the 3 measures that we'll focus on today are treadmill running at 85% VO2 max to fatigue, which was performed before and immediately after the single dive as well as the dive week. A static hand grip endurance at 40% of maximum hand grip strength, followed by a 2-minute occlusion of the exercising arm, which was performed before and after each dive, and a 70-degree head-up tilt for 15 minutes or pre-syncope, which was also performed before and after each dive. And you can see a couple of these tests in the pictures on the right. That far right picture shows an individual on the tilt table being tested for orthostatic tolerance. And the picture on the left shows an individual performing the isometric hand grip endurance test. And the specific measurements we took include treadmill time to fatigue at 85% VO2 max, maximum hand grip strength and time to fatigue at 40% of max hand grip strength, head-up tilt table tolerance and physiological responses to head-up tilt, and cardiovascular variables including heart rate, blood pressure, stroke volume, cardiac output, and forearm blood flow. So in this study, 11 of 12 divers were asymptomatic during the 15-minute head-up tilt, but interestingly, one diver who experienced orthostatic intolerance pre-dive actually improved his head-up tilt times after the dry O2 dives. And then following both single and repeated dives, blood pressure and systemic vascular resistance during head-up tilt were increased post-dive. And the percent decrease in treadmill exercise time to fatigue was much greater after repeated dives compared to the single dive. And these graphs show changes in hemodynamic variables during head-up tilt, and the blue circles represent before diving and the red circles represent after diving. So you can see here on that left panel that systolic and diastolic blood pressure were increased both at baseline and during head-up tilt. And in the panel on the right, you can see that forearm blood flow increased and forearm vascular resistance decreased post-dive, although systemic vascular resistance actually increased after diving. Figure A on the slide shows the comparison of exercise tolerance between previous immersed O2 and air dives compared to these dry O2 dives. And you can see that there's no significant difference in the percent change in treadmill time to fatigue between O2 wet and O2 dry dives. And figure B shows hand grip data, which did not change significantly from pre to post dive or across dive days. So in summary, dry O2 dives did not affect head-up tilt time, except to improve it in the one diver who is predisposed to orthostatic intolerance. Hemodynamic responses to head-up tilt showed a pattern of increased non-peripheral constriction. We also observed that while tilt time was reduced following wet air dives, it was not affected after wet or dry O2 dives. When we looked at dynamic exercise performance, that decreased more after the dive week compared to the single dive in this study, but the decrease in performance was not significantly different from wet O2 dives. Subject-reported symptoms that caused divers to reach volitional fatigue actually shifted from local muscular fatigue to overall central fatigue following the dive week. So in conclusion, repeated exposure to hyperbaric O2 independent of water immersion adversely affects dynamic exercise performance, but not isometric strength. Breathing O2 during repeated dives may protect against diving-induced orthostatic intolerance. So moving on, the next study focused mainly on surface exercise performance following 100% O2 exercise dives. Since many diving environments may require exercise, typically swimming, to be performed during the dive, we wanted to look at these exercise dives to compare to the resting dives. And since previous studies showed a decrease in surface exercise tolerance after resting O2 dives, the purpose of this study was to determine how performance was affected when underwater exercise was included as part of the dive. Again, there were 12 participants whose characteristics are in the table on the right. These dives were conducted in the test pool at 1.35 ATA, breathing 100% O2, and there's both the single dive as well as the consecutive 5-day dive week. During each dive, subjects performed 30-minute work-rest intervals, where work intervals were maintained at a heart rate of 95 plus or minus 5 beats per minute on an underwater cycle ergometer. As before, treadmill time to fatigue was measured before and the day after both the single dive and the dive week, and max hand grip strength and endurance hand grip were measured before and after each dive. And you can see the pictures on the right here showing the divers on the bikes as well as at rest in the test pool. All right, so these graphs show the comparison between resting and exercise dives for max hand grip there on the top row and hand grip time to fatigue on the bottom row. Looking at the exercise dives first, you can see that hand grip endurance there on the bottom right significantly decreased post-dive and across dive days. And then shifting over to the resting dives on the left, you can see a different pattern that pre-dive max hand grip strength was lower than post-dive, suggesting a delayed fatigue effect from the previous dive. But there's no change in endurance time after resting dive days or over the dive week. And then the results for treadmill exercise are shown in these graphs. Figure A shows the change in performance between the single dive, which is in red, and the dive week, which is in blue for each subject. And resting dives are shown on the top and exercise dives on the bottom. Shifting over to figure B, you can see that overall treadmill running time decreased significantly more after the exercise dive week compared to the resting dive week, but there was no difference between resting and exercise dives after just a single dive. In summary, dynamic exercise time to fatigue was found to decrease by 46% following an exercise dive week compared to 21% after a single exercise dive and 28% after a resting dive week. While max hand grip was decreased over the dive week and resting dives, hand grip endurance decreased after each exercise dive and throughout the dive week. From this study, it was concluded that repeat 6-hour dives with intermittent exercise significantly decreased post-dive exercise performance and isometric strength, and the decrements in exercise performance are greater after exercise dives compared to resting dives. Since previous investigations of air dives at rest and exercising showed no decrease in post-dive dynamic or static exercise performance, this finding suggests that it is the combination of O2 and exercise that causes performance decrements. All right, so the third study I'm going to talk to you about looked at the impact of repetitive long duration water immersions on vascular function. And although there's been quite a bit of work done to document immersion effects on autonomic and hemodynamic function, that's a mouthful, as well as change in fluid distribution and excretion, no studies had looked at whether the changes in blood volume associated with post-immersion hypovolemia could affect endothelial function. So the purpose of this study was to quantify changes in endothelial function and vascular stiffness following repeated long-duration dives with surface-supplied air. So for this study, 15 healthy male divers participated in the study. Characteristics on the table on the right, underneath a picture of them relaxing in the test pool. For this study, divers completed 4 consecutive 6-hour resting dives at 1.35 ATA. We used the EndoPAT device to measure peripheral endothelial function, which is represented by the reactive hyperemia index, or RHI, and arterial stiffness, represented by the augmentation index, or AI. AI is measured by pulse wave analysis on the EndoPAT, which is actually dependent on heart rate. So I will also present some results on AI normalized to a heart rate of 75 beats per minute to account for those heart rate effects. Beat-to-beat arterial pressure and total peripheral resistance were also measured at the same time as AI and RHI. And then blood draws were performed before and after the first and third water immersions and at follow-up. And the change in plasma volume from baseline was calculated from hemoglobin and hematocrit. So what we found is that water immersion day had a significant effect on RHI, which was increased by 12.3% on water immersion day 2 and by 16.1% on day 3 compared to water immersion 1. Heart rate was significantly lower post-immersion compared to pre-immersion, but significantly increased over repeated immersions. RHI and heart rate were significantly higher at follow-up compared to baseline, and augmentation index was significantly higher post-water immersion by an average of 33.3%. We corrected for heart rate. That normalized AI was also significantly increased by 10.7% post-immersion. And those results are shown here. RHI is in panel A, heart rate panel B, AI in panel C, and AI normalized to 75 beats per minute in panel D. On the top 2 panels, you can see RHI was significantly increased on both water immersion days 2 and 3, while heart rate was significantly increased on water immersion days 3 and 4. Both were also elevated at follow-up compared to water immersion 1. And on the bottom 2 panels, you can see augmentation index and normalized augmentation index were consistently higher post-water immersion compared to pre-water immersion. As expected, plasma volume was significantly decreased following each water immersion and remained decreased at follow-up testing. Systolic blood pressure significantly decreased, and total peripheral resistance increased post-water immersion. And those significant results are highlighted in this table. So the findings from this study were that RHI significantly increased by the second day of water immersion, but returned towards baseline levels by water immersion 4, which was perhaps a result of increased stiffness and shear stress over that dive week. Arterial stiffness was acutely increased after each water immersion, but this effect seemed to be transient since there was no main effect of water immersion day. We also observed a decrease in systolic blood pressure and an increase in endothelial function, both of which are typically associated with a decrease in arterial stiffness. However, we observed an increase in stiffness in this study. So it may be that the transient increase in stiffness was not enough to counteract that drop in systolic blood pressure from immersion-induced hypovolemia. So we concluded from this study that 4 consecutive 6-hour water immersions caused a transient increase in arterial stiffness following each water immersion and an increase in endothelial function measured by reactive hyperemia index on the second and third days. Changes in systolic blood pressure also diverged from the changes in arterial stiffness, which is a deviation from clinical findings. So future studies in this area should focus on the mechanisms driving an increase in RHI post-immersion, as well as investigate the effects of O2 compared to breathing air. There may be some sort of effective reactive oxygen species breathing O2 versus air. So that would be interesting to look at. And then studies should also investigate the cause of the transient increase in augmentation index following water immersion. So the research presented here was supported by the Office of Naval Research and NAVSEA Deep Submergence Biomedical Development Program. I, of course, have to get the fine print that the opinions and assertions presented here are not those of the authors and not official views of the Department of the Navy or U.S. government. And if anyone has specific questions regarding the Warfighter Human Performance Program, you can contact Dr. John Florian, and his information is shown here. And just to tell you a bit about some current research, the study that I'm working on now is focused on the bioenergetics of the military diving population. And this study will have 3 phases, the first of which is a general characterization of anthropometry, nutritional status, and performance capabilities of various diving populations within the military. The second phase will involve laboratory measurement of energy expenditure during controlled diving operations. And then we'll move out into the field for the third phase to measure energy expenditure during normal diving operations. And from this study, we hope to be able to quantify any energy deficits caused by diving and what their effects might be on performance. And from there, we can explore whether dietary interventions should be implemented to maintain mission and warfighter effectiveness in these scenarios. So that wraps up my presentation of research. As promised, this slide shows various programs having to do with military research for anyone who is interested in that. Personally, I went through the Naval Research Enterprise Intern Program, which obviously shaped my entire career path. And these are summer internships that can be done during or immediately after undergraduate or graduate school, and they are paid, so that's nice. There are also quite a few scholarship programs listed here that will pay for graduate school if you're working in a certain field and then place you in a government service job in that field after graduation. So I will leave this slide up for anyone who wants this information while I take questions. And with that, thank you for having me present today.
Great, thank you, Erin. That was super interesting. We really appreciate your time today. I myself am not familiar with diving. I'm not a scuba diver. So for those of us who aren't as familiar with that activity or this line of research, this is a multi-part question. Can you describe a dive scenario that an actual Navy diver might experience in the line of duty? And then what symptoms do they report following those dives, or what feelings are they reporting, so that we can sort of get a better understanding of what the diver is experiencing?
Well, I can't get into too terribly much detail, but just in general, there are quite a variety of diving specialties within the military. So we have everything from special operations, so SEALs, for example. There are combat divers within multiple different services. There are also search and rescue. There are working divers who might be working on something like ship's husbandry, where they're down working on, the propellers of ships or something like that for repairs. We have instructors. We have Air Force has divers for rescue in case an aircraft goes down. So those are some of the different types of military diving. It can be a variety of just a huge spectrum of the amount of time that they're in the water, the depth that they go to, the temperatures, which is why on one of those first slides I had a list of some of the different scenarios that our program has, that the Warfighter Human Performance Program has investigated. So depending on the length of the dive and depending on the temperature that they've been at, generally it's just that divers report fatigue when they get out of the water. And if anyone has been recreationally diving, that's probably something that they've experienced as well. You just kind of feel really tired the rest of the day. So what this line of research has been trying to parse out is whether that's the effect of a breathing gas, whether that's the effect of just the immersion itself, is it the pressure? And then these future studies will be looking at whether there's an energy deficit associated with that. So I hope that answers your question.
Yeah, great. Thank you. That's really helpful. Another question that came in, do we know if there are any long-term health consequences related with some of these more acute changes that you see in the physiology with these dives, particularly with the sort of heavy diving multiple days in a row?
No. For just studies that we are doing, typically quite a few of these symptoms experienced after that long dive week are resolved, within the next week, if not by the 24, 72-hour follow-ups that we do. Overall, it's really not known the very long-term effects of repeated diving like this. There's some evidence that there could be effects to bone density just because of the extended periods of reduced gravity, but as far as I know, there's not very conclusive evidence on that.
Great, thanks. Another question is, do we know what type of physical fitness, muscular strength, power, endurance, etc., is most associated with successful diving performance?
The diver physical readiness test is typically comprised of a swim, a running time trial, push-ups, pull-ups, sit-ups. There has been some work, and I think there was a presentation for, I believe it was either the Army or the Air Force a few months ago, regarding some new developments in how to get a better measure of readiness. For obviously, it will be a little bit different for divers than other people, which is why pull-ups and the swim have been incorporated into that test. Typically those are, I think that's really the only information that I have on that. I don't know of any direct correlations with specific types of muscular endurance or performance with the diving, in the diving community, and success there.
Okay, great. What about, do we have any idea what the average max VO2 for these divers looks like?
I believe that was on, it's listed on the subject characteristic slides. So here, the VO2 max for that group was 52 plus or minus 7 milliliters per kilogram per minute. Let's go, whoops. So this group was 47 plus or minus 5. And skipped it. And then that one was 51 plus or minus 2. So 40 to 50 range.
Not too shabby, right?
They do a lot of running and swimming, so it is not surprising.
Yeah. So here's another question that came in. Has anyone looked at these same questions using nitrox or heliox instead of 100% oxygen or surface air?
So obviously not in, we have not done that. Nitrox and heliox have both been studied extensively, and there has been some work done on that here at NEDU, typically looking at saturation and decompression, but we have not personally tested that in these scenarios.
Great. How about, has anyone compared bone mineral density between divers and the matched control service members?
No, we have not. One of the things, like I said, that I will be looking at in the bioenergetics study, if I can get to that slide. Oops. So the first phase of this bioenergetics study will be a characterization of quite a few factors, but one of those will be using the DEXA scan to get bone mineral density, bone mineral content, as well as the other measures of body composition. So we will have to do some research to be able to compare that to non-diving service members, but we will at least be able to say what's the average and what's the range of bone mineral density for a variety of divers within the military.
Great, thanks. Another question, this is from one of our staff members. For the 6-hour dives, were the subjects at rest or exercising, and what impact does that have?
Well, I'm not sure which one they're referring to. The first study that I presented, those were resting dives. But the second study that I presented was the 30-minute work rest intervals. So for a total of 3 hours of exercise and a total of 3 hours of rest. And that did impact their performance on land on the treadmill 85% VO2 max test. It did decrease their time to fatigue significantly compared to the resting dives.
Great. Okay, here's a metabolism question. Does the thermal environment and its effect on ventilation affect how you plan to measure metabolism? What is your method of measuring non-aerobic contributions to energy expenditure underwater?
So I'm assuming this is referring to the bioenergetics phase that's coming up. I honestly cannot answer that at the moment. We still have some workup dives to do to determine So we'll actually be using the doubly labeled water method to measure energy expenditure. And we will be at the same time measuring body temperatures at multiple different sites, as well as core temperature, but it will be in thermoneutral water. So we aren't going to be testing the effects of cold water, because obviously that can very much increase energy expenditure if the individual is in cold water. So when we start in the laboratory, everything will be thermoneutral, and moving forward, when we're going out into the field, then we will be able to hopefully associate what the energy expenditure with not only their activity in the water, but also the water temperature itself. I believe there's a second part to that question. I missed it.
That's all right. We've got lots of more questions to get through, so we'll let you slide on that one. If a diver's had a particularly strenuous dive week, are there things that you currently are working on that can help them with their recovery, so that they have quicker recovery time?
Honestly, a lot of what we have been focusing on specifically up until now has just been determining what are the problems that they experience after the dive. And we are starting to move into more of now that we know what some of those problems are, how can we improve that for the diver once they do surface, and improving their recovery. So we do have in addition to the bioenergetics project, there are a couple other projects in the works that will hopefully be able to address some of those specific issues that they have post dive, and figure out a good way to deal with those performance decrements and recover.
Great, thanks, Erin. This question is about the water environment. Are there changes in energy expenditure or bioenergetics between salt and freshwater dives?
That's a good question. Not that I am aware of. There could be some differential fluid shifts. I don't believe that the actual energy expenditure would be different. There may be some differences in how we measure it, especially with that doubly labeled water method, which I will have to look into, because that's not something that I had thought about. So I appreciate that question.
All right, we're going to take a couple more. So what types of physiological adaptations do you tend to see with these divers over the long term, whether because of physical training or occupational training?
I believe one of the big ones is typically an increase in pulmonary function with divers over time. I'm sorry, one second. So I actually have Dr. John Florian here with me, and he has been, like I said, he's the head of our human performance program. So I just would like to ask him real quick, what changes or adaptations over time are there any adaptations over time in divers that you've noticed, just due to their operations and operational requirements and training requirements?
So we haven't done any sort of longitudinal assessment that would probably assess that, but we are going to look at, as we start to develop these baseline piece of information for bioenergetics and photo studies, look at epigenetic changes that occur, look at changes in some of these functions, but in general, pulmonary function doesn't change, I believe. I think pretty much that's just based on normal genetics. I don't think there's going to be any sort of adaptation there. Training can certainly be effective, the fitness level, but as far as things like pulmonary function, and all the other changes.
Okay, so yeah, like I said before with the bioenergetics project, we'll hopefully be able to get some baselines for what a traditional diver, military diver looks like in terms of their performance, in terms of their anthropometry, in terms of their nutritional status and metabolism, but it doesn't sound like there have been really any longitudinal studies of how they might adapt over time to that environment.
If you've registered for the webinar, you will get a copy of the presentation sent to you in a few days. We thank you all for joining, and at this point, everyone here in the national office will also take a moment to thank Dr. Simmons for her time. All right, and with that, we'll say goodbye. Thank you.
Bye, thank you.